Great. Well, thank you for having us. It's a real pleasure to be part of this session. I really enjoyed that last talk about breaking down silos. And hopefully what we can do is convince you that we're taking a similar kind of approach, at least philosophically, for breaking down silos and enabling research within the space of Alzheimer's and related dementias using this project. So, as was said, I'm Mark Cookson. I'm a cell biologist by nature, and I'm co-leading this iPSC Neurodegenerative Disease Initiative, or iNDi, with Michael, who's here from the sister institute, NINDS. And so let's talk about first, in the next slide, what are some of the issues that we know about with iPSC work? So I think one of the key issues, and we talk a lot about the issues around rigor and reproducibility of iPSC work, which is that for the most case, you would take a patient line, so a fibroblast or PBMC from a patient, make iPSCs, and then revert those mutations back to the wild type variant. And you know, of course, that works very well. You have then a pair of lines that you have some confidence we can compare. And there are collections of these in the second half of the slide here. Answer ALS, for example, has something like 1,500 lines collected from patients. NINDS has supported Rutgers to make a catalogue of disease-associated lines that should be available. But of course, there are difficulties with that, right? So the first is that the familial mutations, so those genetic variants that are of large effect size, are relatively rare and often hard to access. Making isogenic lines is not always done, and they're not often quality controlled. Just in the last talk about democratizing research, I mean, something that can get in the way is just legal agreements for sharing. And then I think most importantly for us, it's very difficult to make a comparison between, say, a presennial mutation and a frontotemporal dementia mutation, because they're in different backgrounds from different cells, often made in different ways. And so our approach to doing this was to set up this iPSC Neurodegeneration Initiative, iNDi. And the initial scope of this is to make about 150 or so variants in Alzheimer's and related dementias. So these are defined. So Alzheimer's disease, diffuse Lewy body disease or Lewy body dementia, frontotemporal dementia, which sits on the spectrum with ALS, and then make those all in consistent backgrounds. So the initial set will be the parental line, and I'll talk about that in a minute, both heterozygous and homozygous mutations, but then taking the pathogenic variant and reverting it back to wild type. So the real trio will be having a mutation introduced and a mutation reverted. We'll also make knockouts, some knock-ins for halo tag. And I will say at this point, and in interest of full disclosure, we had 3 external partners for this. So the Jackson Lab have been a major partner, but Synthago also helped us here, and Thermo made some of the lines as well. And we had some redundancy between the different suppliers there. Michael's going to talk a little bit about phase 2. So I'm just going to focus on the first phase of this, which is that engineering phase. How do we engineer iPSCs at scale? How do we generate all of these things in a relatively short order? So in the next slide, so I'm not going to. There's not a lot of data in this talk today. There's a lot of sort of conceptual things. This is probably the most complex thing that we did in the first year, which was to pick some lines that we're going to then use for engineering. And as you can see, we did a lot of quality control and decision making. Some of it was around genomics, so understanding what the genetic background of the lines were, understanding that they didn't carry known functional mutations, but also establishing that we can edit, genotype, differentiate in ways that we think the field would want to use, and that of course they retain pluripotency without any obvious problems. So in the next slide, what we'll show is the first 8 lines that we looked at. And again, the data here isn't particularly critical, but it shows you the things that were most important to us. Using the methods that we use, the methods that Synthago used, the editing actually wasn't really predictive of whether we're going to use a line or not. One of the key things was that the line grew well, but still had an intact P53 system. And so to the best of our knowledge, none of the lines had an oncogenic event. So the one that we've ended up using is a variant of a HIPC line called COLF2, which had a mutation but was then corrected and subcloned. So that's going to be called COLF 2.1. And for the first round of editing, that's the one that we'll use. And we have confirmed that it can indeed, differentiate, it grows well, it edits, and it has no pathogenic mutations. So the next line. So I'm going to hand over in a minute, but I'm going to talk very briefly about what our plans are to distribute. So as you can all imagine, distribution of lines at this scale is really, there's not many people that can do it. We have committed to open science where we can, so the plasmids for this are all available on our gene already. We expect to be distributing trio sets, so again, the parent, the mutant, the revertant early next year, and that will go through Jackson Laboratory again. It'll be very similar to when you order a mouse, so you'll be able to just literally pick it from a web-based catalog. We have also started distributing few lines on an early access basis, and if anybody on this call wants to get access to them, they can contact myself and Michael, and we can tell you what's available, what's being made. We haven't necessarily done all the QC on this, so we're hoping that people can give us feedback and say that they, that it behaved well in their lines. And someone in the question is asking about regulatory consent. This line is, to the very best of our knowledge, free of any MTA issues. It can be shared under a very simple MTA, and we have the full provenance back to the original consent. So there's really very little in the way at this point of distributing this in a way that's completely compatible with legal and ethical constraints. So if we go next slide. I will say just briefly, so I mentioned that we started with Alzheimer's and related dementias, and that was because that's what we were funded for. And that was sort of a set of 150 genomic variants. But once we had the structure up and running, other people saw it and said, well, we'd like to add to it. So with aligning science across Parkinson's disease foundation, we've added about 20 so pure Parkinsonian variants, so without dementia. And through the Chan Zuckerberg folks, we've added 60 neurodevelopmental variants that were not in the initial set. So part of what we're trying to say is, as well as being open, we're also making something that scales well and can be added to. And in fact, as you see here, there's an effort to make really functional lines, so things like DEDCAS9 and the like. So things that can be then used as tools, but again, in the same genomic backgrounds. So I think with that, one more thing on this early access. So our measure of success here, I was really interested to talk about measures of success in papers in the last talk. Our measure of success here is that people use the lines. That's the key thing. And we've made 150 projects approximately so far, and those are out in 50 labs in 10 countries across 3 continents with no limitations. So I think this is what success looks like for one of these democratized projects. So with that, I'm going to hand over to Michael, who's going to give you some, really specific examples of how this is working for us.
Great, thanks so much, Mark. So I'm going to start off here by just telling you about 3 vignettes about some of these users of the early access lines, because this is just something that Mark and I are very proud of, that people are already starting to adopt these and use them in their own research. And then tell you a little bit about phase 2 of iNDi, which is really this foundational data generation of phenotypes that ensue in disease-relevant cell types generated from iPSCs that harbor these mutations. So the first vignette is from Gujan Bu's lab at Mayo Clinic, and they're very interested in ApoE4 biology. So they've requested the entire suite of ApoE mutant lines and performed karyotype analysis. It was all normal, and markers of pluripotency, which were again all consistent with a pluripotent iPSC line, differentiated these successfully into 3 germ layers, and they're now well on their way toward generating organoid models of Alzheimer's disease with these different ApoE4 alleles or ApoE alleles. Next set would be from Erica Holzbauer's lab at the University of Pennsylvania. They're really interested in vesicular transport, in particular neuronal autophagy, which is quite a bit different from autophagy that goes on in other cell models. They differentiated COLF 2.1 iPSC line into neurons with the neurogenin system, and then they performed time-lapse microscopy of retrograde moving autophagosomes here. And actually, it was really neat because they looked at the control parental line and then a LRRK2 mutant line. And you can see here in this chymograph of this normal gradual retrograde transport of LC3 positive autophagosomes in the control neurons. But in this LRRK2 mutant, you can see how these autophagosomes just freeze in place. This is a phenotype that had been reported in a different iPSC mutant line with this same mutation. So it was really gratifying to see that they could replicate this exact result in the knock-in line from the iNDi set. And this was work that was done by Dan Duo and Erica's lab. And then the third set, and I just think this experiment is really, really cool. Is performed by Deborah Kronenberg-Versteeg. And she has this elegant model in which she takes iPSCs, human iPSCs, differentiates them into microglia, then depletes microglia from organotypic brain slices, either from mice or from humans, from intraoperative specimens obtained from human patients. She gets rid of the endogenous microglia, and then she puts these iPSC-derived microglia on top of the slices, and what happens is those iPSC microglia invade, because there's no resident microglia anymore. She wiped them out with antibodies, and they ramify gorgeously. To me, somebody who's looked at a lot of microglia, I've never seen any iPSC microglia that look this beautiful. And this is being done with the COLF 2.1 line in these human brain slice co-culture. So what we've seen and heard from investigators who've requested these is that it looks like the COLF 2.1 line is going to work for a lot of different applications, differentiating into neurons, into glia, making organoids. I think this line is going to be a workhorse for the field. So we really couldn't be happier about how the community is adopting this. In the last few minutes, I want to tell you about something that's really exciting that's happening on the NIH campus. And I think actually, in many ways, is a parallel to a lot of fantastic work that Devin Krogan just described about what's happening at UCSF and Gladstone. And that is, how do we start to understand ramifications of these disease-related mutations on biology, and do this in a scalable manner, and do this in a multimodal fashion, where you're not just looking at one type of data, but you're looking at a concert of different kinds of data sets that play well together. So what we've done is we've begun to develop a series of technologies that will let us differentiate these different mutant iPSC lines at scale. This is a completely roboticized process. It's being carried out by a Hamilton robot, which is set to arrive in the new building for CARD in February. We're going to be performing differentiation of these iPSCs at 48 batches at a time into neurons, and then followed by a set of high content microscopy, functional synthetic lethality, CRISPR-I screens, total RNA-seq, and proteomic mapping, not just of the proteome of the cells that harbor these mutations, but also APMS-based strategy with halo knock-in lines of wild-type and mutant proteins. Once these data sets are generated and validated with QC metrics, they're going to be uploaded into an open data repository on the fly, searchable by the field. And we're going to build this data repository in such a way that not only can people explore the data that's in there, but they can use this data as cross-referencing data sets against their own data sets that they generate in their lab. So we're very excited about this project. I'm going to show you some of the tools that we've been building that I think is going to enable this. First of all, here's just an image of what this robot's going to look like. That's going to do the automated cell culture. Quite similar in many respects to the Hamilton robots that the New York Stem Cell Foundation has successfully used in their own high-content iPSC culture. We've modified a version of our neurogenin differentiation protocol that can be actually transfected into cells with an automated lipofectamine stem technique. It's a piggyback-mediated transposon that can integrate TET-on neurogenin as well as selectable markers. And this allows for a robot to generate these things en masse. It doesn't require safe harbor engineering and picking clones. This is something that can be done in a polyclonal population. So really amenable to high content approaches. This is an image of the Colt 2.1 line with the piggyback neurogenin differentiated from iPSCs into glutamatergic neurons. We're developing these protocols as we speak and refining them. These are going to be accessible to the community in protocols.io, and actually we should be releasing this one for the piggyback version pretty soon. All the plasmids, as Mark mentioned, are being deposited onto AdGene. So these neurogenin neurons from Colt 2.1 map, as we would expect, to fetal-aged excitatory neurons shown in these UMAP projections here. This is done by single-cell sequencing. So we think we're making relevant cells. They're not age cells, but then again, this is iPSC cultures. We don't expect that. And then we've invested a lot into automated proteomic workflows here. A lot of the stuff that groups do is easy to do when you're doing one set of proteomics experiments, but once you start to scale this up and you're pumping out samples from a robot, you need a different approach. So actually, this is a lot of help from Evan Krogan's group in terms of getting an automated sp3 protein extraction technique up and running on a Kingfisher instrument. Now a human won't need to touch the samples as they roll off the robot, go directly onto this Kingfisher instrument for sp3 protein extraction, and this goes into thermo scientific Eclipse mass spectrometers with Thames Pro and data independent acquisition mass spec. These methods look like they're revealing far more peptides than the more traditional methods that we were using previously here, just with whole cell proteomics. And you can actually look and see as iPSCs differentiate into neurons and their complement of proteins. We're going to be using day 28 iPSC neurons. And the traditional method by hand, you can see the spread on a PCA plot here across samples. It's much tighter with this fully automated Kingfisher sp3 method that we're going to employ at scale. And then the next is something I'm particularly excited about. This was really work that grew out of a collaboration with Martin Kampmann at UCSF that enabled us to, for the first time, do genome-wide CRISPR inhibition screens in iPSC-derived glutamatergic neurons, the same neurons that we're going to be making in INDi phase 2. One takes these iPSCs that we introduced neurogenin and DCAS9 technology with piggyback, then overlays these with a genome-wide guide library, differentiates these into neurons, and basically counts guides at the beginning and the end of the experiment to find out the effect of gene knockdown on neuronal fitness. When you do this in a test case by knocking down a neurodegeneration-related gene, TP43, one can actually then interrogate buffering genes and synergistic lethal genes, very, very similar to the synthetic lethality screens that were done in yeast very successfully, but in this case in disease-relevant iPSC-derived neurons. Now, this initial technology wasn't really scalable for a robot. It required 100,000 different guide RNAs in this second-gen library. So we've gotten access to a third-gen library from Jonathan Weissman that's reduced the size of the library from 100,000 guides to get genome-wide coverage down to 20,000 guides. So this lets a whole genome screen with 1000x coverage being done in 20 million cells. That's something that can be done on a single 15-centimeter dish. And so when we look at the performance of this single guide library versus dual guide library, we actually find that in the neurons, the dual guide library is outperforming the single guide library, despite the fact that it's 1/5 the size. You're getting larger effect sizes for each targeted gene with this particular library. So I think that now we're at the scale of being able to do this genome-wide fashion with automated robotic production of the neurons. And then finally, this high-content microscopy. We're using a super res Sora spinning disk with an auto plate loader. This is very similar to what Steve Finkbeiner is doing at Gladstone. We're applying a cell painter type approach that Ann Carpenter pioneered at the Broad. Here's just some images of mitochondria from these Colt 2.1 neurogenin neurons. Here we can make out the very fine detailed structure of these organelles. So this is going to be a multi-organelle high content imaging platform to generate data for artificial intelligence interrogation. So with that, I'd like to thank the team. It's really been amazing. Andy Singleton is the leader of the Center for Alzheimer's and Related Dementias, who really championed this project and allowed us to get the necessary funding to launch this. Having deep collaborations with a number of other groups at the NIH, which includes Data Technica International, led by Mike Knowles. For us, Fagri has been really intimately involved in a lot of the CRISPR-I work here. Lirong Peng in the single cell work, and then an amazing team that we've assembled for INDi itself. Erica Flores has been doing all the cell culture, Andy Key leading all the mass spec work, Dan Ramos doing the functional genomics, and then Marinita, who actually isn't on the slide, but leading the high content microscopy. All of the engineering was done by Synthico partners as well as JAX The JAX team, led by Bill Scarns, just one of the most delightful people I've ever collaborated with. And then CZI has really been an essential partner here. Mark Sutherland, Katja Bros, and Alden Connor. We've had great input from collaborators in Cambridge, Florian Merkel, Andrea Yang, and actually a lot of the nuts and bolts of this project were worked out with a long-term collaborator and colleague of mine, Bruce Conklin at Gladstone. So with that, if we have any time, happy to answer any questions.